Whilst we are addressing a very, very abysmal HDMI cable here, I would like to say that the only reason why I stand here before you today is because I didn't really work directly with Brian, but I represent the interface between the end of the official Brian era and the start of the unofficial Brian era in around 2004. And so the key reason for actually standing before you is on the following slide, which is, it would be a little unfair to say that Brian has not had a direct impact on my life. I was appointed in 2004 to the Powderjack University Lectureship in Biomedical Engineering. It was actually the first appointment in Biomedical Engineering in Oxford. Then in 2006, I found myself being appointed to the Bellhouse Foundation University Lectureship in Biomedical Engineering. I was served as a mentor of the first postdoctoral Bellhouse Fellow at Magdalen College. But I think my most daunting role was actually the one that Jamie beautifully described. And I should emphasize that Jamie and I have not actually seen each other in several years, so there was no joining up of our presentations. But what actually happened is that I took over from Brian as tutor and postdoctoral fellow at Magdalen College. And Jamie, I know it was meant as a compliment, but you could not have put it more succinctly. It was made blatantly clear to me in my first 2 years that all I could aspire to be, potentially, someday, equally good of a tutor. I actually spent my first 2 years, there's actually another student here from that era. Charles, please make yourself known. I'm delighted we were able to get the 2 students who were taught by Brian into the room. And so I kept asking for feedback as to how I was doing. It was made very clear to me that there was a certain Brian-ness that I could not quite catch. I was trying to figure out what that Brian-ness was. And sometimes it was inspiration, sometimes it was contextualization. And so one day, it was actually in Jamie's 3rd year as an undergraduate, I thought maybe I've cracked it. And I'll show you something which I have kept. And I don't know if you remember this, Jamie. So this is actually, and I'll read the note to you. Constantin, looks like you're right about that limits thing. Should have listened to you. Or maybe I've made a mistake. Anyway, kept me amused for 15 minutes while I worked in the bar. Jamie. So Jamie and I had a debate about spherical polar coordinates during one of our tutorials. He had walked away unconvinced and had actually spent his time serving other morning bars. He didn't have anything else to write on. So this is actually Timrol, on which this mathematical proof has been written. And I thought, if I could send him out into the bar, worrying about polar coordinates, I must have done something.
I have no idea on the first.
So that's quite all right. We'll do it without slides.
I'm sorry.
That's quite okay. The research that Brian inspired in me was actually inspired by the entire medical engineering unit. So my first laboratory in Oxford was at 43 Banbury Road. It was in 2004. Mark was there, George Costigan was there, Roger, a much beloved MEU technician who many of you remember was there. What I remember about this building was there was a whole bunch of people who were wondering about how to optimize transport across the skin. That was actually the key problem. But what I found absolutely fascinating when I landed there is that all of them, without exception, were doing it in a multidisciplinary way. And it's fair to say that drug delivery research in general was a little bit of trial and error. There hadn't actually been a huge amount of proper engineering methodology put behind it. And what I discovered when I arrived at the MEU is that not only were there people there who were bringing together concepts of fluid mechanics and heat transfer and material science and were talking to immunologists, they were also worried about building prototypes, comparing modeling and theory to experiment. And that was a very important working ethos to land in as a new academic in Oxford. I didn't know anything about transdermal delivery, had never worked in transdermal delivery, but my interactions with the people at the MEU at the time inspired me to start working on transdermal delivery. And what we actually did was we started thinking about ultrasound mechanisms that could be used to push things through the skin. So as it turns out, when you propagate ultrasound through the body, as Jamie showed, you can actually get tiny bubbles appearing and collapsing. And it's a bit like opening a bottle of sparkling water 1 million times a second. So Brian and others' ideas was to actually use a pressure wave and a jet to actually push these particles through the skin. Our idea was actually to use these tiny bubbles that could then start oscillating 1 million times a second And as they oscillate, they start transferring momentum onto their surroundings, and therefore can actually entrain particles in them at depth within the skin. And so we've now had 4 generations of students, and actually the latest generation is sitting right there. Her name is Anne-Charlotte van Blokland. Actually work on transdermal drug delivery problems that have been inspired by that time at the MEU. But those transport mechanisms are actually pervasive throughout medicine. So delivering drugs isn't just a problem in skin, it's also a problem in tumors. It's also a problem in cardiovascular disease. And so we're able to take that concept that started at the MEU and show that these tiny bubbles that are actually pulsating can also superbly deliver ultrasound and drugs better into tumors. So when you have a drug circulating in the bloodstream, only 1% of that drug circulating in the bloodstream will actually reach the target tumor. And even that 1% will only be within 50 microns from the nearest blood vessel that delivered it. But there's a cancer cell over here, 200 microns away, that will never, ever see any drug. And this is because that molecule is finding it just as difficult to travel from the vasculature to that cell as it's finding to cross the stratum corneum. So the problem fundamentally is exactly the same, and the solution potentially is exactly the same. So research-wise, what has happened since is we've actually been able to produce a very, very large cohort of students. We're actually funded as a National Centre for Drug Delivery Devices. It's a £25 million endeavour today, looking at all the research contributions that have been put together, working with the pharmaceutical industry to bring these physical-based approaches for transport of drugs, be they ultrasound waves, magnetic waves, shock waves, rockets, together with modern drugs, biologics, vaccines, antibodies, mRNAs, to see how these approaches can be combined to do more with existing drugs. To actually stop developing new molecules, stop making molecules bigger, but actually take the ones we have and just deliver them better in the hope that they can actually achieve the therapeutic potential. And I think very much of that was actually inspired by that initial interaction. There is at least one slide I'd like to try and show you, and I very much hope that it will let me. Gary, are you able to do anything at all with the computer?
Try it.
Okay, so I have, and I'll be happy to share it with you afterwards. So there are really 3 things about where we are today, and Lionel very kindly left it to me at the end to talk about where we are with biomedical engineering today. So when I arrived in Oxford in 2004, biomedical engineering was actually the product of 3 big successes. The biggest one by far was Powderject. There'd also been Mirada Solutions, and of course the Oxford Knee. These were the 3 pillars: imaging, drug delivery, orthopedics. I absolutely loved my time in the medical engineering unit. Gary, are you all right?
Just recognize, sorry.
So we were all at the medical engineering unit, and it was a wonderful building. It had windows that opened, which I have to say is a gloriously recent feature. It had daylight, it had tall ceilings, but it also had suspended floors, which meant that when we're trying to do ultrasound or multiphoton imaging measurements, every time a bus drove by on the Bangalore Road, we had to start over. And the one memory I have is we had a fantastic DPhil student called Maynard, who was trying, I think, to build a soup-up version of a Powderject device. And he used to run these large explosions in the basement. And we would stand by the window and watch all the tourists hit the floor, particularly the Americans, every time an experiment was done. So it was fantastic, and it was a great, but it wasn't exactly a 21st century environment for biomedical research. And I think there were 3 things that Brian really felt during his time here. He felt he would very much like to have had better access to clinical expertise and to a more biologically suited environment for translating this research into the clinic. He wanted to preserve the multidisciplinarity, and he felt that the next generation really deserved proper facilities in order to realize some of the things he had wanted. And then there is one 4th thing which was the fact that if we measure the success of the IBME today, on the top right, always, it says, since 2008, we have started 13 spin-out companies. Now, that's a great number in itself, but the only reason why that metric is even there is because of someone like Brian. Because when I got to Oxford in 2000, 2001, it was still a swear word to have the temerity to have a business as an academic. And so much of what we are today, so the Institute today has raised over £50 million of funding. It has graduated over 320 PhD students. We've had 65 patents granted. We have initiated 32 technology-led clinical trials that would not have existed if it weren't for physical scientists working on a medical site. We have started 13 companies. And I think these three things that we are, between 2004, the snapshot of the MEU, and this extraordinary 3,500 to 4,000 square meters of dedicated physical sciences space on the Churchill Hospital site, were a chemical reaction that requires many ingredients, but like all chemical reactions, it required one catalyst. There can be absolutely no doubt that that catalyst was Brian. And for that, we are all eternally grateful to him. Thank you.